All right, so today we're going to be talking about protein synthesis a bit more in depth than we did last time. Last time we just talked about RNA, its structure, what it looks like, what it's made of. We talked a little bit about the overall protein synthesis process using this image, but today we're going to talk about it more in depth. So, protein synthesis. DNA molecules provide the information for making proteins. However, since DNA is too big to leave the nucleus, DNA gives its message to mRNA. And that RNA is going to be the instructions that are used to build the protein. And the workers for all of this are going to be different types of RNA molecules. This whole process of going from DNA to RNA to protein is called protein synthesis. So going through the three types of RNA one more time. There are three types that are going to help out. There's the mRNA or messenger RNA, and this is the type of RNA that is going to go from the DNA in the nucleus out to the ribosome, carrying the DNA message. That's why it's called messenger RNA, because it carries DNA's message. The second type of RNA is ribosomal RNA, and it helps build the ribosome. And it reads the mRNA message. So our RNA or ribosomal RNA helps make the ribosome, and it reads the mRNA. And the third type of RNA is transfer RNA or tRNA. And tRNA are going to bring in amino acids to help build the protein. So we're going to look at how all of this works a little bit more in depth. The protein synthesis, the process of building a protein, has 3 steps. Step 1 is called transcription. I spelled that wrong. So transcription. In transcription, DNA is rewritten into a molecule of mRNA so that the RNA can leave the nucleus. So step 1, transcription, rewriting DNA as RNA. Step number 2 is going to be translation. And that is going to be translating the mRNA code into amino acids. Remember, amino acids build proteins. So many amino acids linked together make a protein. So translation is the process of making amino acids in the right order from the RNA. And then step number 3 is going to be the protein folding. So once we have all the amino acids linked together, the protein's going to fold up, and then it can finally do its job. So we're going to look at those 3 steps one at a time. So step number 1 is transcription. Transcription is going from DNA to RNA. So transcription is the process that happens within the cell nucleus. This happens in the nucleus because DNA is in the nucleus. DNA to RNA has to happen where the DNA is, which is in the nucleus. And enzymes are going to take DNA and read it to make a molecule of mRNA. The mRNA is built by the enzyme RNA polymerase. Now, the RNA that is made here is going to be the complete information for building a protein. The reason that we have this step is because DNA cannot leave the nucleus. DNA cannot leave the nucleus, so we have to make a molecule that can. So DNA is going to be rewritten as mRNA because mRNA can leave the nucleus. Now, the way that this works is actually quite simple. what we're going to use are our base pairing rules. Now remember, DNA had the letter T, but RNA has U, and our base pairing rule is U goes with A, and G goes with C. This is for mRNA. So when we see the letter T, T goes with A goes with U, C goes with G, G goes with C, C goes with G, A goes with U, C goes with G, A with U. We have 3 T's in a row. Remember, T's go with A's. A's go with U's. C's with G's. G with C. T goes with A goes with U. C goes with G. G goes with C. I'm going to have to scroll up. C goes with G. G goes with C. Twice there. So when you're doing these, all you need to remember are your base pairing rules. A goes with U. C goes with G. The only reason that people get tripped up with this one is sometimes when they see the T, they're like, well, what does that go with? Remember in DNA, T goes with A. So when you look at your mRNA, you should see no T's but U's. So that was step 1. You rewrote the DNA as a molecule of mRNA, and that is called transcription. One way that you can remember that is transcription. Script means to rewrite. So to transcribe means to rewrite. We wrote it both using letters. So we rewrote it in a different way. So that was step 1. Step number 2 is translation. Translation is going from the RNA to the protein. So we're going to use the RNA that we made here in step 1 to make the protein. So translation is converting information in the mRNA into the language of a protein. It's called translation because we're kind of changing languages here. We're going from bases to amino acids. They're going to look different. This is going to take place at the ribosome, which is located in the cytoplasm of a cell. And remember, proteins are made up of amino acids. So this process is going to involve taking the RNA and creating a series of amino acids. Now, there are 20 different amino acids out there. And the way that it's converted from bases to amino acids is by using what we call the genetic code. Now, the genetic code is going to be the same in all living things. All living things have the same genetic code. And the genetic code is going to consist of 64 codons. So you can think of that as 64 different codes. A codon is a set of 3 bases that are going to represent one specific amino acid. And all organisms are going to use the same genetic code. So an example of this would be the 3 bases UAC and mRNA code for the amino acid tyrosine. But it doesn't matter if you're a bacteria or a human or a bison or a tree. If you have the letters UAC, that codes for tyrosine in every single organism. The difference between organisms are the way in which these amino acids are linked together. So there might be a different order of amino acids, they're going to be mixed around, and that's going to be what makes us different, is the order of the amino acids. The code itself and how it's read is always the same. So I feel like the best way to understand translation is to just give it a go. So here, this was our DNA. We took our DNA and we rewrote it as mRNA in the process of transcription. Now what we're going to do is take this mRNA and split it into codons. Codons are groups of 3. So when I do this, I always take my group and I split it into groups of 3. Each group of 3 is 1 codon. So there is my RNA split into groups of 3. And in translation, the codons are read by the ribosome in these groups of 3. So it will read AUG, then it will go CGU, GUA, and so on. Now, one thing to know is that codons do not just code for amino acids, they also provide instructions for making the protein. So, we're going to have special codons that code for start and stop. So the stop codons mean, okay, stop reading, protein's done, we're done, stop here. And those codons, you don't need to remember these, they're always going to be written on the chart for you. We'll have a chart. UAA, UAG, and UGA are the stop codons. So anytime you run into one of those, it means stop. Stop what you're doing, you're done. Protein's finished. And we're also going to have the same for start codon. The start codon is always AUG. AUG means, hey, start here, proteins needs to be made. Start reading here. That's where you start doing the process, and then you stop when you get to a stop. Now, one thing to take into consideration is that the start codon always codes for the same amino acid, methionine, meaning all proteins start with that same amino acid. So when we have a sequence of DNA, you don't start reading until you get to an AUG, and you don't stop until you get to one of the stop codons. And it tells you where a protein is going to start and where it's going to end to make it nice and easy. So again, you'll have a chart and it'll tell you start, it'll tell you stop. So you'll know this, you'll be able to recognize it when you read the chart. Now you might be wondering how our amino acids are going to get to the ribosome. And that happens by using tRNA. So remember, tRNAs are going to transport the amino acids to the ribosome. Each tRNA has its own amino acid that it's responsible for because of its structure. And this is done through base pairing. So tRNA carries only one amino acid, and that amino acid is identified based on its anti-codon. So here's our tRNA here. Let's say we have an mRNA, and hang on, I'll just make my own instead of using that one because I can't read what that one says. We'll just make my own. So let's say we have our RNA here. And my RNA codon is going to be AUG. The ribosome is going to read this. It's going to say, okay, AUG. tRNAs are floating around, and each tRNA is carrying with it an amino acid. Like this. And the tRNAs are going to pop in, and tRNAs have 3 letters on the bottom. Each tRNA has a different 3 letters. And they're going to pop in and out until there is a match. So when they match up, so A goes with U goes with A, C goes with G. When they match, it's going to be like, oh, you belong here. This amino acid will be dropped off. And that is how they know where to put each amino acid. So if there's not a match, it's just going to pop out, and they just keep randomly circulating until it finds the perfect match. When it finds the perfect match, they say, okay, this amino acid belongs here, and the amino acid will be dropped off. And that is using the anticodon. So here's our codon, and this is the anticodon. And this is how we're sure that we're putting our amino acids in the correct place, because they have to bond together to work. So a little bit, and we just talked about the tRNA structure. tRNAs kind of look like the letter T. Sometimes you'll see it looking like a lowercase t, sometimes you'll see it looking like an uppercase T, but it's always going to look like a T. The bottom of the T has 3 letters, and those are called the anticodon. The anticodon is going to pair up with the codon. When those two match perfectly and they can bind together, then the amino acid will be dropped off, and that's how we know it happens in the right spot. So the steps of translation. The ribosome is going to read the mRNA codons. When it finds the start codon, it starts building the protein. tRNAs bring in amino acids. They know they're in the right spot when they match based on the anticodon. The amino acids are linked together to build a protein. And then when we hit the stop codon, the process stops. I'll post some videos of how this process works using a video on YouTube. That way you can kind of see it happening, because I can't really show the movements of one molecule in and out until they match on here. So there will be some more explanation videos on Canvas. Right now I want to walk through how you read this chart when you're building your protein. So here is the DNA that we had. Using transcription, we rewrote the DNA as an mRNA. We separated the mRNA into groups of 3 called codons. They're color-coded. And now we're going to do the process of translation. And translation is the process of building a protein by taking these codons and turning them into amino acids. Now, when you do this, you'll always have a chart. And here's what the chart looks like. I always like to write first. At the top, I like to write second. And on this side, I like to write third. So the way this works is you look at one group of 3 at a time. So here's my first group of 3, A, U, G. My first letter is A. So over here where I wrote first, I find the A. Here's the A. That narrows me down to this section here. So just this section, I can ignore the rest of it. So all of this I can ignore. Can ignore all of that. My second base is U. So up here where I wrote second, I find U, and I follow my U down. Follow it down into this box here. That means I can ignore all of this. My third letter is G. So I find the G on the third column side. So there's many G's here. There's G here, 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 and here. Now I've already marked out this section so this G doesn't work. I marked out this section, so this G doesn't work. The bottom G, I marked out that section, so that G doesn't work. The only G that is left is this one here in the box that I've already circled. I follow that one over, and I see a UG right here, and it stands for MET. Now, if you look at this chart, you might have said, well, Miss Ray, I could have just searched all over the chart and found the 3 letters because they're all written here. And I would say, yes, that is true. On this chart, it is written right there, AUG. You could have just searched and found it. However, that's not always the case. A lot of the charts that you're going to have don't always have those 3 letters together like that. Sometimes it'll just have MET sitting right here, and you won't have the 3 letters there. So it's very important that you understand how to read the chart in the event that the groups of 3 are not already on here. So I'm going to go through it a few more times. So we did our first one. Our second group of 3 is CGU. So my first letter is C. I find it over here on this side. Here's my C. I'm left with this section. My second letter is G. I find G at the top. Here's G. I follow it down, and they overlap right here. My second letter is U. So I find U in the box that I've already circled. Here it is, CGU, and that stands for ARG. Now remember, MET and ARG, those are both amino acids. So we're currently building the protein. All right, so looking at our next group of 3, GUA. I find my G on this side. I'm down to this box here. U I find at the top. They overlap here. Find my A on the third side and follow it over. Here's GUA. And my letters are VAL. All right, so keep going through. If you want to go ahead and pause, figure out the others. While you do that, I'm going to slowly work, and then you can speed up and catch up. I'd like for you to try this a little bit on your own. So go ahead and pause and figure out the rest of the amino acids. All right, so go ahead, check yours against what we have here. There will be lots of practice on this, so don't worry. There will be lots of practice for that. All right, so now we're down to step 3, which is protein folding. So we made our protein. Here's our protein. The next step is for the protein to fold up. So amino acid chains will become proteins, active proteins, only once they are freed and folded into their 3D shape. Proteins can't do their job until they're folded up into their 3D shape. Now, these enzymes, or these proteins, could become enzymes. They could become cell or tissue structures. So proteins could be many different things. It could be the protein that gives my eyes the color. It could be an enzyme that helps break down food. It could be many, many, many different types of things. Remember, everything in our body is controlled by protein. So your hair color, the functioning of your digestive system, everything is controlled by proteins. Proteins do the main job in our bodies. So there are many, many different things that proteins could do or become. So here left, we have one more practice section. So we'll go through this as a whole. All right? So we have our DNA. The first step in making a protein out of our DNA is to do transcription. Transcription is when we rewrite our DNA into mRNA, and we use the code A goes with U, C goes with G. So I see a T, I put an A, I see an A, I put a U, C, G, G, C, C, G, A, U. So you can finish that out. Our next step is going to be put our mRNA in groups of 3. Those groups of 3 are called codons. And then we are going to translate. I'm going to look these up in our amino acid chart to figure out the amino acids that they stand for. So I know that AUG is MET and CGU is going to be ARG. And you can keep doing that on and on. And that is how we go through this process. So there's going to be some practice on protein synthesis on Canvas. So go ahead,